The first activity that we're going to use is activity 14, and that's creating something called a Z projection. So taking all of that 3D data and compressing it into a 2D image for either a lab presentation or to print out and put into your lab book. So for all of the 3D data, we'll be using one of two files, either the omx3ddata.tiff or the kidney3ddata.tiff file. So let's drag and drop one of those in. We'll see that I've already created these as RGB files. These have 3 channels in there, so we've got the blue, green, magenta setup. Hopefully you guys would know how to get to this point. And this is a Z-stack running through a kidney tissue section. So the problem is, how do we present all of this data in a 2D format? So to do this, we're going to go to Image, Stacks again, and we're going to go to the Z project function. Alternatively, we also have a stack shortcut up here, which we can also use to select Z project. This gives us a dialog box saying, do we want to include all of the slices within the data, and also a projection type. And what we have to think about when we're looking at this is that the different projection types might work differently or better for different types of image samples. The best way of working this out is to go through and just try them one by one and see which one you think presents your data the best. So what we're looking at is each pixel down through Z. If we're using average intensity, then the final image that's plotted will have an average of all the intensities that are running through there. So in this example, there's quite a lot of black backgrounds or out of focus from kind of image 26, between 1 and 26. So if we used average here, it might not work very well. We can always go in and say we want to start at image 27, and let's use, for example, the maximum intensity. Okay, so that's a representation of all of the data that's included within that Z stack. So the activity here is to go through and play around with the different types of Z projection techniques and work out which one you think works best for presenting the data for the kidney section and also for that OMX3D data.tiff image as well.